Hi, this video will explain about oxidative phosphorylation, still under cellular respiration. Oxidative phosphorylation will be an explanation about electron transport chain and chemiosmosis. The learning outcomes for this lesson is for students to be able to illustrate, to explain electron transport chain, which is the pathway of electron transport involving an ADH dehydrogenase, ubiquinone or coenzyme Q, cytochrome C reductase, cytochrome C and cytochrome C oxidase. Next, students should be able to explain chemiosmosis, proton motive force, and the last one is to explain complete oxidation of one molecule of glucose in active cells to produce 38 ATP. First, why do we need oxidative phosphorylation? Because through substrate level phosphorylation, during glycolysis and Krebs cycle, we only produce 4 ATP per glucose. It is only a small amount. We need more energy. But most of the energy extracted from the glucose molecule are stored in NADH and FADH2. So the NADH are produced from the glycolysis, link reaction and Krebs cycle, while FADH are produced from the Krebs cycle only. So the energy in these two molecules need to be harnessed to convert into ATP. Okay, it needs to be, how do we harness the energy? Okay, by using the electron that they carry. So the electrons within the NADH and FADH2 must be collected through electron transport chain along with the chemiosmosis. So the energy released by the electron transport chain will be used to synthesize the ATP during chemiosmosis. So electron will flow from one component to another component through the chain, here the ETC. So during the transportation of the electron, there will be energy release. So the energy will be used to bind phosphate to the ADP. So it will produce ATP. So let's look at the detail. So this is the diagram that shows ETC here. This is the component of ETC. This is the component of ETC. And this is where chemiosmosis occurs. Electron transport chain and chemiosmosis. Okay, oxidative phosphorylation involves these two processes. These two processes occurs in the inner membrane of mitochondria. So this is the membrane, the inner membrane of mitochondria. The phospholipid bilayer of the inner membrane. So these are all proteins that are embedded in the. These are all protein complexes that are embedded in the phospholipid bilayer. Remember, the proteins embedded in the phospholipid bilayer have several functions. One of them is protein, some of them are enzyme. Okay, so first you need to know is the currency, meaning that one molecule equal to how many ATP. We use the analogy as currency. Contoh $1 USD equal to berapa ringgit Malaysia, kan? Okay, so one NADH will able to produce 3 ATP, while one FADH2 will be able to produce 2 ATPs only. So what is electron transport chain? It is a series, do not forget the word, series of electron carriers, because the electron will be moving from one molecule to another molecule to the next molecule in the chain. Okay, so it involves the electron carriers. So these are all electron carriers embedded in inner membrane of mitochondria that carry electron from NADH, starts from here or starts from here, through a series of redox reactions to synthesize ATP. So redox reaction means should involve oxidation and reduction. Okay, so releasing electron means the molecule is oxidized. Receiving electron means the molecule is reduced. One molecule release, one molecule will be accepting it. Location of the electron transport chain in the inner membrane of mitochondria, including the cristae. Cristae is the folded part. Okay, this is the inner membrane, the folded part. The folded part is called cristae. So along the inner membrane, the cristae is also actually the inner membrane, but it is the part where it is folded. Electron transport chain also involved in photosynthesis, which is located at the thylakoid membrane of chloroplast. So we will learn later on. We will find the same thing. We will come across the same thing when we are discussing the photosynthesis. Okay, let's look at the electron transport chain. The details will be explained in this chapter. The purpose of electron transport chain first to break a large free energy from the glucose into a series of smaller steps through electron transport chain that release energy in manageable amount. We cannot release release it and then the energy is like a bomb. So we need to harness it bit by bit. Okay, smaller steps. So it needs a smaller step. This energy will be used to form ATP. The basic concept of electron transport chain. The hydrogen atom from the NADH and FADH2 dissociates into protons and electrons. Okay, the dissociation occurs in the mitochondrial matrix here. Okay, so the hydrogen ion and the electron will be separated. The proton, which is the hydrogen ion, will be released into the matrix of mitochondria. Okay, so here an NADH hydrogen plus will be released into the matrix, while the electron, okay, I will use a red color, purple color. Okay, the electron will be passed to the electron transport chain. That's why the name is electron transport chain, not hydrogen transport chain. So then the H plus, the proton, will be pumped into the intermembrane space here. Because once it is released, it will increase the concentration of hydrogen ion. So an NADH release the hydrogen, then the hydrogen dissociate into proton and electron. So the electron will be passed to the ETC, while the hydrogen ion, the proton, will be released into the matrix. So the concentration of the hydrogen ion will increase in the matrix of mitochondria. So this will induce a facilitated diffusion from the matrix of mitochondria into the intermembrane space. Okay, this does not need energy. This occur passively. Kan, dah belajar tentang transport across membrane, betul tak? Pergerakan, the movement of molecules from higher concentration to a lower concentration. That is a through facilitated diffusion. So the electron enter transport chain, electron transport chain, then during which the energy will be released during the transfer of the electron. So the energy is released, then it will be used to synthesis ATP by oxidative phosphorylation. So the synthesis of ATP is done by an enzyme. So this is the enzyme ATP synthase. So how does the energy release is used? Will be explained later. First, we need to memorize the name of the component of the electron transport chain here. Let's look at the next slide. Okay, the component involves seven substances. The first complex is complex 1, an ADH dehydrogenase. Complex 1 is an enzyme named an ADH dehydrogenase. So from the name itself, we know that the substrate is the NADH. And what does it do? It dehydrogenate. It removes hydrogen from the NADH. Okay, complex number 2 is the succinate dehydrogenase. The substrate is the succinate, and what does it do? Remove hydrogen from the succinate. Okay, actually from the Krebs cycle, remember FADH involved during the formation of succinate from succinyl CoA. From succinate to fumarate. Succinate to fumarate. Succinate is oxidized to fumarate. So succinate release hydrogen to FAD, reducing FAD to FADH2. Then succinate is oxidized to fumarate. So succinate dehydrogenase. The third component is the coenzyme Q, or also known as ubiquinone. This is a coenzyme, not a protein. The fourth component is the complex 3. So complex 3 again is a protein or enzyme, to be specific. The name as cytochrome C reductase. So what does it do? It reduce something. Okay. And the component number 5 is cytochrome C. So this enzyme actually involves this component. Okay, we will see later on. And component number 6 is cytochrome C oxidase. What does it do? It oxidizes. This one, reduce. Okay, the name itself helps us to know what they do. Still involve cytochrome C. One cytochrome C reductase, complex 3, complex 4, cytochrome C oxidase. It does have to do with the redox reaction. And the last component is the oxygen atom. Okay, complex, look at the yellow highlighted sentence. Complex 1, 2, 3 and 4. 1, 2, 3 and 4 are integral proteins, meaning that the proteins are embedded in the phospholipid bilayer and they are not motile, they cannot move. So electron carrier, the components alternate between being oxidized when they lost an electron, or they will be reduced when accepting electron. So this is called redox reaction. Okay, electron carrier of the chain is reduced when it accepts electron from its uphill neighbor. Look at the graph here. This is a graph actually. The free energy increasing, right? So the substances, the component here, meaning this one, and ADH, has a higher energy level than FADH2. FADH2 has higher energy level than complex 3. NADH, this is NADH, this is complex 1. Okay. NADH has higher energy level than complex 1. Complex 1 has higher energy level than complex 2, and then FADH, and then complex 2. Then complex 2 higher energy level than complex 3, and complex 3 higher than 4. So NADH and FADH also has different energy level. That's why NADH able to pass the electron to the first complex, while FADH2 unable to pass to the first complex. Instead, it will pass the electron directly towards the complex number 2. Let's look at the first step in the electron transport chain. In step 1, it involves complex 1, the NADH dehydrogenase. Complex 1 is a flavoprotein which has prosthetic group flavomononucleotide, and also iron sulfur protein. So you don't have to remember this. But what we need to know is NADH dehydrogenase, the complex 1 will receive electron from the NADH. So NADH either comes from the glycolysis, or from the liquefaction, or also from Krebs cycle. So NADH carry the electron from these processes and transfer the electron to the NADH dehydrogenase, the first complex. So NADH dehydrogenase is reduced because it accepts 2 electrons from the NADH H+. Remember, it involves 2 hydrogen atoms, which will then dissociate into proton and electron. So NADH plus H+ will be oxidized, forming NAD+. So what happened to the NAD+? It will return to the either Krebs cycle or the Lin reaction. The pathway of electron from FADH2. You see, because I've already mentioned before, an ADH from the glycolysis, Lin reaction, and Krebs cycle. It has higher energy level compared to FADH2. So an ADH able to transfer the electron to the first complex, while the pathway from FADH2 is different from the electron carried by an ADH. FADH2 will transfer the electron, passes the electron to the succinate dehydrogenase, which is the complex 2. So succinate dehydrogenase is reduced and FADH2 becomes oxidized. The electron carried by FAD has lower energy than electron carried by NAD+. So remember, FADH2 are released or produced from Krebs cycles. Thus, 1/3 less energy is released than the oxidation of NADH. Step 2 involves ubiquinone. So ubiquinone is here. It is a mobile carrier. Because complex 1, complex 2 is non-mobile. Coenzyme Q is small lipid molecule. That's why we can see that it is embedded in the lipid layer, lipid bilayer, not the phosphate bilayer, not the phospholipid bilayer. Not it is because it is a lipid molecule. Jadi sebab tu dia duduk dalam region lipid. So it can move from here to here. So it is a mobile. So it receives the electron from FADH2 and pass the electron to the complex 3. So the NADH dehydrogenase returns to its oxidized form because it passed the electron to coenzyme Q. This from the pathway of NADH. Right. electron from NADH and FADH2. If the pathway is from FADH2, the coenzyme Q will return the succinate dehydrogenase into its oxidized form, because the coenzyme Q received the electron from the succinate dehydrogenase. Okay, so it gets electron from both parties, the complex 1 and complex 2. So coenzyme Q is reduced and then it will be oxidized when it passes electrons to cytochrome C reductase. Here the complex number 3. Okay, in step 3, the cytochrome C reductase, because it involves cytochrome C and it will reduce the cytochrome C. How does it do it? Because it will pass the electron to the cytochrome C, thus reducing the cytochrome C. When it releases the electron, it will be oxidized. Cytochrome C reductase has heme group as prostatic group, so it can accept electron from the coenzyme Q and then oxidize when it passes electron to the cytochrome C. Cytochrome C is also a mobile carrier, but it is a protein type, while ubiquinone is a lipid type. Cytochrome C is a protein type. So cytochrome C will be reduced when it accepts electron from the cytochrome C reductase or complex 3. Then it will pass the electron to cytochrome C oxidase. Hence the name oxidase, because when it receives the electron from cytochrome ni, kompleks 4 ni, bila dia terima elektron daripada cytochrome C, automatically it will oxidize the cytochrome C. So that is why its name is cytochrome C oxidase, because its function is to receive the electron, meaning that it is oxidizing the cytochrome C. Complex 4 will be reduced when it accepts electron from the cytochrome C and oxidizing and it will be oxidized when it passes electron to oxygen molecule, which is the final electron acceptor. So oxygen is reduced when it accepts the electron from the cytochrome C oxidase and hydrogen ion from mitochondrial matrix. So because of the binding of these two, or the exception of these two proton and electrons, water will be formed. That's why in cellular respiration there are production of water. Okay, proton pumps. Proton pumps, the energy released during electron transfer in electron transport chain is used to pump the protons from the matrix into the intermembrane space of mitochondria. Okay. So the pumping of the proton, increasing the concentration of hydrogen ion in the intermembrane space. So the increasing of the hydrogen ion concentration in the intermembrane space will induce a movement of hydrogen ion into the matrix of mitochondria. Okay, back into the matrix of mitochondria. Because proton is accumulated in the intermembrane space, so the concentration is higher, causing proton gradient across the membrane. This is called as proton motive force, the proton gradient. So the proton motive force will induce chemiosmosis to occur. So what is chemiosmosis? It is the movement of hydrogen ion from high hydrogen ion concentration to low hydrogen ion concentration through an enzyme named as ATP synthase to catalyze the synthesis of ATP from ADP and inorganic phosphate. Okay, what happens? The gradient of the gradient of the proton cause the hydrogen ion to move back into mitochondrial matrix down the concentration gradient through ATP synthase. So the movement while the hydrogen ion is moved through the ATP synthase, okay, the flow provides energy to generate ATP like a turbine. Okay, macam empangan hydro kita kan, the movement of water menggerakkan turbine and then produce energy. So this energy generated from the movement of the hydrogen ion, the flow of the hydrogen ion, will generate energy used to bind phosphate inorganic to the ADP, producing ATP. Okay, remember the gradient induced flow of hydrogen ions provide energy to generate ATP. So accordingly, an ADH plus H plus produce 3 ATP, while FADH2 produce 2 ATP. Okay, that's all explanation about oxidative phosphorylation, which involve electron transport chain and chemiosmosis. Okay, about the shuttle, this is an extra reading. You can read. Anything that you don't understand, you can ask. But it is not. It will not be explained in details. Because it is out of syllabus, but if you want to ask, you can ask. I will answer the question. Okay, so that's all. Thank you.